In the last two videos, we've seen that electrophysiological recordings and optical imaging techniques are extremely useful in measuring neuronal activity in an awake, behaving mouse. These measuring techniques allow us to correlate neuronal activity with behavior. But in order to get a causal argument linking that neuronal activity to a specific behavior, we need to be able to manipulate those neurons. We need to be able to stimulate them and inhibit them with high specificity in terms of time and space, and see how that impacts the other network effects and the behavior that we're interested in. Until recently, to stimulate neuronal activity, the best tool that neuroscientists have had is to put stimulation electrodes into the brain. And by driving brief current pulses through those stimulation electrodes, one could activate the neurons close to that stimulation electrode. However, axons of passage, or neurons in connected brain areas, would also be stimulated, limiting the specificity of electrical microstimulation. On the other hand, to inhibit neuronal activity, neuroscientists have had even poorer control. Typically, they've lesioned brain areas in animal models, or they've injected pharmacological agents like tetrodotoxin to block action potential firing, or CNQX and APV to block glutamatergic synaptic transmission. These reagents certainly block activity near to the injection site, but they do so on long timescales, typically involving many minutes, and of course they don't have any cellular specificity. They inhibit everything nearby, and typically many different types of neurons are intermingled, and so again there's a lack of specificity. Over the last decade, neuroscience has gone through a revolution, allowing us to deal with causal interventions in the mammalian brain through genetically expressing actuators that control neuronal activity, and so that allows us the specificity of mouse genetics to decide which cells we're stimulating or inhibiting, and to have these actuators under optical control. And that means we can point light at specific cells that might express our optogenetic actuator and then take control of that cell, for example, making it depolarize and fire an action potential. And we can then utilize the specificity of light, being able to turn light on and off with millisecond temporal resolution, and being able to point it at specific parts of the brain in order to take a highly specific control of the mouse brain during behavior. Let's see how that works. The optogenetic revolution begins with the cloning and identification of light-activated cation channels, and in particular the identification of channel rhodopsin 2 as a light-activated cation channel in this seminal paper here from Nagel et al. In 2003 began the optogenetic revolution for the neuroscientists. Channel rhodopsin 2 is a transmembrane protein that was cloned from the green algae Chlamydomonas reinhardti. It's a plasma membrane ion channel, and bound to it is the molecule retinal, which is an aldehyde derived from vitamin A, and that's covalently bound to the channel rhodopsin protein. And it's the retinal itself that interacts with light. And so retinal is a relatively small molecule. It absorbs a photon, and that then drives a conformational change in the retinal, changing it from a trans to a cis motif, so it changes its structure, the retinal. The retinal is bound into the channel rhodopsin channel and causes a conformational change of the channel protein and increases its open probability. And so in that way, light directly gates the cation channel encoded by channel rhodopsin 2. So we've already seen other ion channels that are gated, for example, by neurotransmitter like the glutamate receptors, or by voltage like the voltage-gated sodium channels. Here we now have a light-activated channel where absorption of a photon on the millisecond timescale causes an increase in open probability. And it's a relatively non-selective cation channel, so it's permeable to potassium, sodium, calcium, and also protons. And under physiological conditions, the major conductance is an inward sodium and calcium current that then has a reversal potential of somewhere around 0 millivolts. So in terms of putting it inside neurons, this is an excitatory light-activated channel. The next important step in the optogenetic revolution was to put channel rhodopsin into neurons. And that was successfully done by Ed Boyden when he was in Carl Deisserud's laboratory in the seminal paper from 2006. What they found was that they could express channel rhodopsin in cultured hippocampal neurons in a dish. And the cells that expressed channel rhodopsin turned out to be light sensitive, and brief pulses of blue light would drive action potential firing with millisecond temporal precision. So they gave 10 millisecond light pulses, and action potential firing in cells that were expressing channel rhodopsin occurred with very low jitter and highly reproducible. Every time there's a blue light flash, an action potential was driven in these neurons. And they found that they could drive the neurons at different frequencies. So they could stimulate at 5 hertz, at 10 hertz, and even at 20 hertz. And they could reliably drive action potential firing with every blue light flash. This is remarkable because there were many things that were uncertain. It was unclear, for example, whether retinal would be sufficient in mammalian neurons to allow the single expression of channel rhodopsin to be sufficient to drive neuronal action potential firing. And so this caused the beginning of a revolution, and it's really the robustness of the channel rhodopsin that's made it used widely across the neuroscience community. An important question, of course, was how well channel rhodopsin 2 works in vivo in the living mammalian brain. And so here we've expressed channel rhodopsin 2 in the primary somatosensory cortex. We come in with a whole cell membrane potential recording from one of the neurons that's expressing channel rhodopsin. And in vivo, there are membrane potential fluctuations that are large in amplitude and occurring on a variety of timescales. And so it's interesting to see to what extent we can get reliable control of action potential firing under these conditions of high variability in vivo. And it turns out that at least under some experimental conditions, brief blue light flashes applied to cells in vivo cause extremely reliable action potential firing, just like it had been described in the cultured hippocampal neurons by Ed Boyden. So, irrespective of the membrane potential at the time of stimulus, each time a blue light flash, here 3 milliseconds in duration, was delivered to the cell, it would cause a depolarization, it would reach action potential threshold, and an action potential would be evoked. This occurred in response to every blue light flash, but there was some variability in the timing of the action potential with respect to the onset of the blue light. As you'll see in the membrane potential, there are spontaneous fluctuations in the pre-stimulus membrane potential. So sometimes the blue light is delivered when the neuron is hyperpolarized, and at other times the blue light is delivered when the neuron is spontaneously depolarized. And it turns out that that makes a difference to the precise timing of the action potential. If the neuron is spontaneously hyperpolarized, the blue light comes on, we need to depolarize by a long distance, and that takes some time before we reach action potential threshold. On the other hand, if we're spontaneously depolarized, we turn the blue light on, the depolarization begins at the same point, but now there's less depolarization that needs to happen before we hit AP threshold, and the action potential therefore occurs earlier. And so every blue light stimulus gives a single action potential output, but the precise timing varies by a millisecond or so, depending upon the exact depolarization, the state of the cell that we're recording from. And so channel rhodopsin 2 turns out to work extremely robustly in vivo. A next question one might ask is whether stimulation of neurons with channel rhodopsin 2 can drive a behavioral output in the mouse. And an obvious place to look then for optogenetic influence on behavior is by stimulating the motor cortex. So we investigated here in a system that we've already studied in some detail, the somatosensory whisker signaling pathway, where if we stimulate a whisker, we can image neuronal activity across the dorsal surface of the neocortex. with voltage-sensitive dyes. We stimulate the whisker, and the first thing that happens, if we look in the neocortex, is that there's depolarization and sensory processing in the primary somatosensory cortex. Some 5 to 6 milliseconds later, there's an additional hotspot of activity, sensory processing in the motor cortex. And so we can localize the whisker motor cortex in the mouse through voltage-sensitive dye imaging. We can then express channel rhodopsin specifically in this area of the brain by injecting a virus to express channel rhodopsin in the whisker motor cortex. And then in our head-restrained mice, we can then turn on the blue light and see what happens when we stimulate the whisker motor cortex. And perhaps not surprisingly, this causes the whisker to move, and we get into this what appears to be something like the exploratory movement of the mouse as it's exploring its immediate environment, the whisker going backwards and forwards rhythmically as we maintain the blue light stimulus here on the motor cortex area. This occurs with very short latencies and is extremely robust. Every time you turn the blue light on, about 20-30 milliseconds later, the animal begins to move its whiskers in this exploratory way, searching for something in its immediate environment. And so channel rhodopsin 2 works in vivo. It can drive action potential firing, and it can also drive behavior, here visualized in the whisker motor system. A major drawback of the way in which we were stimulating the whisker motor cortex with channel rhodopsin was that we don't know exactly how many cells or exactly which cell types it is that we're stimulating. We're simply applying blue light to the whisker motor cortex, and many neurons, thousands of neurons, were expressing channel rhodopsin, and probably subsets of those were firing action potentials. In order to get to that level of detail where we can stimulate individual neurons, we then need to start putting light beams on specific cells and stimulating those. We've already seen that it's difficult to focus blue light into the brain because the brain is a highly scattering medium. We also saw, when we were thinking about high-resolution imaging, that two-photon excitation offered an interesting way out for fluorescence imaging. And we can use exactly the same phenomenology of two-photon excitation of channel rhodopsin to get optical Z-sectioning and get around the scattering problems of the brain. So if we imagine that this is the energy diagram of the channel rhodopsin, and so we have a ground state, and normally we would give a single blue photon to activate the channel rhodopsin, we can instead of the blue photon deliver two infrared photons, go through the virtual state, so now we have two low energy photons, and that's good in itself. They scatter less inside the brain, so we've got a better chance of focusing our infrared beam into the brain. And in addition, we have this nice phenomenon, this nonlinearity of two-photon excitation, where in order for us to go through this, these two photons need to arrive at the fluorophore within a femtosecond of each other, so we need a very high density of photons in order to get the two-photon activation, and that's only present here at the center of the focal spot, and these other areas, as we try to focus this beam of light into a small area here, has a low photon density, and that's then insufficient to be able to drive two-photon excitation. And so we can confine the excitation of channel rhodopsin through two-photon phenomena, and we can imagine that we express channel rhodopsin in, say, all the cells of a given brain region, and then we simply point our laser beam at different neurons, and we excite them through two-photon excitation, the specific neurons in specific temporal sequence that we want, and we can then gain control at a single cell level with millisecond temporal resolution. This is still largely being developed, but it's clear that this can work if the conditions are sufficiently well controlled. And so there's a great deal of hope for the future that this type of methodology, two-photon excitation, will allow us to take control of neurons and neuronal networks at the required level of single cells and millisecond temporal precision. Remarkably, in addition to the optogenetic tools for stimulating neuronal activity, optogenetic tools for inhibiting neuronal activity have also been developed over the last decade. The first protein that was identified is a so-called halo rhodopsin molecule, which encodes a light driven chloride pump. Absorption of a photon into halorhodopsin moves one chloride ion from the outside to the inside of a cell, and so that causes a hyperpolarization of the neuron. Another molecule called archirhodopsin, or ARCH, is a proton pump, and absorption of a green photon by ARCH removes a proton from the inside of a cell and moves it to the outside, again causing hyperpolarization. For each yellow or green photon absorbed by these pumps, one ion is moved. And so it's clear that this is a much less efficient process than opening of an ion channel, where you can imagine that a single photon causing the opening of an ion channel lets many ions through. And so there's a drop in efficiency, and in general, of course, pumps are known to be much slower than ion channels. And so there's considerable excitement about the recent development of a light-activated chloride channel through genetic engineering of the channel-rhodopsin ion channel. The normal channel-rhodopsin 2 ion channel has some negative glutamate residues sitting here along the pore-forming region, and that's then presumably the reason that it's a cation-selective channel. The cations like to interact with the negative charges. What these two papers managed to do was to convert the cation channel of channel-rhodopsin into a chloride channel, and in part this was done by putting positive charges here in the pore-forming region, and that then allows chloride ions to permeate, and they then made a light-activated chloride channel. And it's possible that this is going to be a much more efficient way of inhibiting neurons compared to the light-activated pumps. The light-activated pumps do nonetheless work to some extent, at least in some cell types. And here's an experiment in which halorhodopsin, the light-activated chloride pump, is expressed in a specific type of GABAergic neuron in the neocortex, one that we've already talked about. This is the somatostatin expressing GABAergic neuron, and what's special about these cells is that they have a very high input resistance, something like 200 megohm, and that means that small currents in these cells create a big difference in membrane potential. And so even though the halorhodopsin may not be the most efficient way of delivering an optogenetic inhibition, it nonetheless worked very well in this cell type. And so here, in an awake behaving mouse, the somatostatin cell is firing away during normal quiet wakefulness. We turn the yellow light on, that activates the light-activated chloride pump, and it causes an important hyperpolarization of the membrane potential and a complete shutdown of action potential firing. The light goes off and action potential firing returns. The inhibition can last many seconds, and for this particular cell type, it's complete. However, it has to be said that halorhodopsin hasn't worked equally efficiently in all cell types, and so it's good that further genetic engineering goes on to improve the efficacy of the optogenetic inhibitory molecules. Indeed, optogenetic tool development is something that's ongoing in many neuroscience laboratories. Researchers are working on making higher conductance ion channels that would allow greater ion permeability and thus a more potent optogenetic actuator. Or they're trying to change the ion selectivity, making calcium permeable light-activated channels or potassium selective ones, in addition to improving the chloride selectivity of the newly developed ion channels. Changing the kinetics is also extremely interesting. Making the channel rhodopsin faster would be something extremely helpful. Right now, if one gives a 1 millisecond pulse of light, the channel rhodopsin turns on rapidly, but it also stays on for considerably longer than one might want. And so it might be nice to have optogenetic actuators that followed the light pulses more accurately. And so faster off variants of channel rhodopsin would be extremely welcome. It's also interesting to think about slower variants that would stay on for longer periods of time, where one pulse of light would leave it on for a long period of time. And indeed, switchable channel rhodopsins have already been developed, where one color of light would turn the channel rhodopsin on, and a different wavelength of light would turn the molecule off. Further spectral variants are being developed so that channel rhodopsins are not just activated by blue light, but by red light, infrared light, ultraviolet light, blue light. And you can imagine a day when there will be different types of channel rhodopsins expressed in many different genetically defined neurons, and we'll be able to separately control their activities through different spectral variants of channel rhodopsins. One can also imagine targeting the channel rhodopsins or other optogenetic activators to different neuronal compartments or subcellular compartments that might all provide interesting information about how neurons function. Finally, we've been discussing optogenetic tools for changing membrane potential, but there are many other cellular and molecular processes that are open to optogenetic actuators, including gene expression, synaptic transmission, and a whole variety of other signaling pathways within neurons and cells in general. So in this video, we've seen that channel rhodopsin 2 is a powerful tool for the neuroscientist. Channel rhodopsin 2 encodes an intrinsic light-activated cation channel that, when expressed in neurons, allows us to control their firing with millisecond precision. This then allows us to ask causal questions about what the stimulation or activity of a specific group of neurons that are genetically defined contributes to a given behavior. We've also seen that new optogenetic actuators are being developed to inhibit neurons with high temporal precision and specificity. And further optogenetic tools are being made to inhibit neurotransmission or to affect different signaling pathways. So altogether this week, we've seen that there are interesting and useful ways in which we can measure and manipulate the mouse brain activity in awake behaving animals. And what we'll look at next week is how we can use these tools to address specific questions relating to sensory perception and sensory motor integration.